Okay, happy Wednesday. We are on to lecture 30 today, but as always, I want to start with a quick review of what we were going over previous lecture, which was lecture 29 on Monday. Monday's lecture was on titration, which you may have already seen in your 1AL class, but whether you've seen it before or not, know that titrations are used to determine the amount of acid or base in an unknown solution. So what you do to set up a titration experiment is you measure out some volume of the unknown, in this case acid solution, and you put it in a flask, and that flask goes below a burette, which is basically a big graduated cylinder with a little valve at the bottom that lets you start and stop the flow of what is in the cylinder. And what you put in the cylinder, in this case, is a precise volume of base with a known molarity. So by combining this known concentration of base with this unknown concentration of acid, you can figure out the concentration of the acid by monitoring the neutralization reaction. The graph that you use to monitor a titration experiment looks like this. It is known as a titration curve. And so you start out over here on the left with 0 moles of base having been added so far. So that's just the initial unknown acid solution. In this case, we have 200 milliliters of it, and we measure its pH, and we find out that it is somewhere around 3. And then we go ahead and open the valve on the burette, and we see that the pH starts to rise a little bit as we add our first, say, 10 milliliters of sodium hydroxide base. This makes sense because the hydroxide is reacting with some of the H plus in solution, which encourages more of the weak acid to dissociate. So the equilibrium shifts towards the conjugate base of the weak acid. So our pH is going to rise a little bit, but it does so pretty slowly as we add more and more base until all of a sudden we've added some magical amount of base that causes the pH to shoot up really quickly. And this vertical part of the titration curve indicates that we have reached something called the equivalence point. And the equivalence point is when the number of moles of OH minus that we've added is equal to the initial moles of HA in that very original acid solution that we had at the beginning. So the fact that these 2 moles are equal to each other at the equivalence point lets us do a little calculation. So we can first find the moles of OH minus by multiplying our known molarity of sodium hydroxide by the volume that we've added at the equivalence point. In this case, that's 100 milliliters, which corresponds to 0.1 liters. So we find that we have added 0.01 moles of hydroxide, and that is how many moles of HA we had in our original solution by definition. So then to get the concentration of the original acid solution, we just divide that number of moles by the volume that we measured out. We know we had 200 milliliters of the acid HA originally. That corresponds to 0.2 liters. And so 0.01 divided by 0.2 gives us 0.05 moles per liter of HA solution. We know that HA must be a weak acid, not just because it says it on the slide, but also because if we just blithely take the negative log of this initial HA concentration, we get 1.3. And 1.3 is not the pH that we initially measured. We initially found that the pH was equal to 3. And so we must be thinking, well, not all of that HA is actually dissociated into H plus and A minus. A lot of it is staying as HA. So that is the hallmark of a weak acid, one that is incompletely dissociated in water. But our takeaway from this particular situation is that we can use this nice handy equation at the equivalence point, that the volume of acid times the molarity of that acid is equal to the volume of base times the molarity of that base. Typically, the part that we don't know is the molarity of the acid, and so we can rearrange the equation to solve for that. Another special moment in the titration of a weak acid is the moment at which you are halfway to the equivalence point. So here our equivalence point occurred when we had added 100 milliliters of base. So halfway there would be at 50 milliliters of base, and we can measure the pH at that point and find that it is 4.74. That pH is a useful value to us because, as we saw last time, the pH halfway to the equivalence point is the pH at the moment that the moles of HA is equal to the moles of A minus, because you have converted half of that HA into A minus. That has special implications for our acid dissociation equilibrium constant expression, Ka, which, as you're probably bored with me saying by now, is equal to the product of H plus and A minus concentrations divided by the concentration of HA. When the concentration of A minus and HA are equal to one another, those terms cancel out, right? It's like putting 1 over 1 or 400 over 400. And so this equation simplifies down to just H plus. If we take the negative log of both sides of that equation, we see that the negative log of Ka is equal to the negative log of the concentration of H plus. We already know that we have a special name for the negative log of the concentration of H plus. That is called pH. And the negative log of anything is P, that thing, pKa, for example. And so here, by measuring the pH at the halfway point, we are also measuring the pKa. And this is very useful because if we have the pKa, then we can just do 10 to the negative pKa and get our Ka value back. And we love knowing the Ka of our acids because that is often indicative of what the identity of that acid is. Ka values tend to be unique. And so if we don't know what this acid is, knowing its Ka value can be very useful to us. So at the half equivalence point, pKa is equal to pH. To kind of summarize the three main parts of a weak acid titration experiment, you have the acid solution all by itself at the start of the titration before you have added any base. But remember that you can't just take the negative log of that acid concentration to get to the pH. You're going to have to use the Ka to figure out what that pH is. So you'll need to solve this equation here for X, and X is your concentration of H plus. That's what you can take the negative log of. How are you going to know the Ka of this acid? Well, if you know what the acid is, you can look it up. But if you don't, you can use the pH at the halfway to the equivalence point. Remember that at this moment, H plus concentration is still greater than OH minus concentration. You're not at the equivalence point yet. But that H plus concentration is going to be just equal to Ka at that moment because HA is equal to A minus. Those two terms cancel. And so taking the negative log of both sides allows us to determine the Ka from the pH. So you can use that Ka to calculate your initial pH if you like. And then finally, at the equivalence point is where our moles of HA are equal to our moles of OH minus. But the equivalence point for the titration of a weak acid has one important difference from the equivalence point in the titration of a strong acid, in that the pH at the equivalence point is not equal to 7. It is not a perfectly neutral solution. When you titrate a weak acid, you are making the conjugate base A minus, and that little conjugate base is a little bit basic. It can react with water, as shown in this equation here, to make HA and a little excess of hydroxide, OH minus, that will lead you to a basic pH greater than 7. So our key differences between a titration of a weak acid versus a strong acid is this slightly basic pH at the equivalence point, the fact that the halfway to equivalence point tells us the Ka of that weak acid, and the fact that we have to do a few extra steps to determine the initial pH. This all leads us right into lecture 30, that's today's topic, on the topic of buffer solutions. So buffer solutions are made out of a weak acid and its conjugate base, and they are very useful to us because they are very resilient. They resist changes in pH when a strong base or strong acid is added to them. And so what we'll be talking about today is how do you make a buffer solution, how might you choose the appropriate buffer solution, and then how does it actually work? How do these buffers resist changes in pH? We ended last time looking at two solutions that had the same pH of each other. Over here on the left, we have a very dilute hydrochloric acid solution. That's a nice strong acid. All of it is dissociated, but because there are so few moles of it, it ends up with a pH of 3.5 nonetheless. We can also make a solution of acetic acid, CH3COOH, that has a pH of 3.5, but we just have to use a lot more acetic acid to do so because its dissociation is not as strong. So here, the difference in concentrations is making up for the difference in the Ka values. So if you wanted to make a solution that has a pH of 3.5, here are two different ways to do it. But if you want that solution to keep its pH of 3.5, there are very big differences between how these two solutions behave. Specifically, let's look at the acetic acid example for now. So here we have three different solutions of acetic acid where I don't just have the acetic acid, I've then also added an equal molar amount of sodium acetate. The acetate anion in sodium acetate is the conjugate base of acetic acid, CH3COO-. And because I've added equal moles of each together, we see very, very little dissociation, right? In each of these pictures, we just see one little H+ circle all by itself. And because there's the same number of H+ circles in all three solutions, we end up with the same pH, 4.7 every time. In this case, the pH value does not depend on concentration, but rather on the fact that we have equal moles of acetic acid and sodium acetate. The reason there's so little dissociation here is because acetic acid was a weak acid to begin with, right? We have this nice low Ka value, and so not very much of it was dissociated anyway. But then when we added acetate to the solution, that's a perturbation to an equilibrium. You've added more product, so the equilibrium shifts towards the reactants by Le Chatelier's principle. And so we have even more acetic acid in solution in its associated form than we would otherwise. And so here we have a way to keep a pH constant even at different concentrations. We can make a similar set of three different concentrations of solution that maintains a different pH of 6.4 by simply using equal molar amounts of a different weak acid and its conjugate base. In this case, our weak acid is carbonic acid, H2CO3, and its conjugate base is the bicarbonate anion, HCO3-. And because carbonic acid has a different Ka value, the pH value that is exhibited by this conjugate acid-base pair is different. So we find that the pH value of these equimolar solutions depends on the conjugate acid and base pair that you use to make them. Specifically, the pH value for this 1-to-1 equimolar buffer solution is equal to the negative log of Ka, or the pKa. So here we are exploiting that halfway to the equivalence point moment from our titration lecture to make these 1-to-1 buffer solutions where the pH sticks at the pKa value. The buffer solutions that you make by adding together equal large amounts of both the weak acid and its conjugate base can resist large changes in pH upon the addition of a strong acid or a strong base. The way this works is by looking at this acid dissociation equilibrium that we have looked at so many times now. If you imagine adding a little bit of strong acid, so adding some H+, well, that's going to shift this equilibrium towards the reactants, increasing HA and decreasing A-. But assuming the concentrations of HA and A- were initially very large, that little shift will probably be comparatively small. Similarly, if you add a little bit of strong base, that strong base will react with H+, decreasing the concentration of one of your products. So Le Chatelier's principle will shift this equilibrium towards the products, making more A- and depleting HA. But if you have a lot of HA and a lot of A-, that little decrease and that little increase will not be a very large change. And this means the pH does not change very much either. So let's look at that. You've seen this equilibrium constant expression for acid dissociation a million times now, but here it is one more time, H+ times A- divided by HA. Indeed, this A- is the conjugate base I keep talking about, and HA is the conjugate acid. We can solve this equation for H+, since the pH is what we are really interested in. And we see that H+ depends on the Ka times the concentration of the conjugate acid divided by the concentration of the conjugate base. But if those two numbers are large enough that the little changes to them don't really matter, then we're in a situation where HA and A- are always about equal to one another. These two numbers are equal, this term is basically equal to 1, and so we end up with the concentration of H+ equal to Ka, or the pH equal to the pKa. And since the Ka for a given acid at a given temperature is always the same, we have created a solution where the pH is basically always the same. You can create buffer solutions of different pHs using weak acids of different Ka values. So the one that we've just explored is acetic acid. If you combine it in equal amounts with its conjugate base acetate, then you end up with a buffer solution pH equal to the pKa of acetic acid. The Ka of acetic acid is 1.8 times 10 to the negative fifth, so the pKa is 4.74, and that is roughly the pH that your buffer solution will maintain. If you instead create a buffer solution out of formic acid and its conjugate base formate, well, formic acid has a Ka value of 1.8 times 10 to the negative 4. That is a slightly larger Ka value, and so the pH is buffered at a slightly lower value compared to acetic acid. Contrast this with a buffer solution made out of methyl ammonium and its conjugate base methylamine. Well, the Ka value of methyl ammonium is 2.5 times 10 to the negative 11th. That is a very small Ka, and so this buffer solution will maintain a very large pH of 10.6. I do want to emphasize that these buffer solutions need to be created using a weak acid. Yes, you can still get a pH of 4.74 by using a very dilute strong acid, but this pH will not be very easy to maintain. So if you use a very small amount of hydrochloric acid, for example, you can obtain a pH of 4.74, and if you add just a little bit of H+ to it, that's going to increase the H+ concentration, but not by very much, so your pH stays about the same. That's good. But the very dilute HCl solution will not be very able to resist changes in pH if a base is added. If you add hydroxide, that's going to react away the H plus in your dilute solution very quickly, even if you add a small amount of hydroxide. There will be some left over. And when you have excess hydroxide, your pH of your solution is greater than 7, in this case 9.26. And that change from 4.74 to 9.26 is huge. That is not an effective buffer. On the molecular level, what's happening here is if we imagine a 120 milliliter solution of HCl that's very dilute, so has a concentration of 2 times 10 to the negative 4 moles per liter. This one's slightly different from the solution on the previous slide. Its pH is 3.7, but the same idea applies. If you add even a small amount, just 40 milliliters of dilute 1.2 times 10 to the negative third molar NaOH, all of those small numbers of H plus cations will be reacted away by the OH minus anions to form water, and then the small excess of OH minus anions will give you a relatively high pH. So even though you were able to achieve the pH you wanted initially, the pH changes a lot after the addition of the base. Contrast this to the same pH as before, so here we're back to the 4.74, achieved using a high concentration of acetic acid and its conjugate base acetate. Because you can make this pH of 4.74 with really high concentrations, then these little perturbations to the concentrations aren't going to matter as much. Sure, you add a little bit of H+, that's adding a product and so shifts the equilibrium left towards the reactants. Yes, you will have more acetic acid than before and a little bit less acetate than before, but these are small changes on top of large initial concentrations, so the pH is about the same. If you were to add OH minus instead, that will deplete the H plus product and the equilibrium will shift towards the right, producing more acetate and depleting some of the acetic acid, but once again, these changes are small compared to the large initial concentrations, and so the pH does not change very much. We can also look at the molecular level changes that were just explored on the previous slide. We are starting from our high concentration but equal moles, 1, 2, 3, 4, 5 acetate ions and 1, 2, 3, 4, 5 acetic acid molecules. When we add 0.2 moles of HCl, what do we see? Well, we have a few more, 1, 2, 3, 4, 5, 6, 7 acetic acid molecules and 1, 2, 3 acetate ions. This happened because the H plus from the HCl increases the products, and so Le Chatelier's principle predicts that the equilibrium will shift towards the acetic acid molecules, which is indeed what we see. Now the concentration of CH3COOH is greater than the concentration of CH3COO-, and our pH is a little bit lower, but not much. We can also consider starting from the same buffer solution, but instead adding a strong base, for example, 0.02 moles of sodium hydroxide. What do we see after those strong base moles are mixed in? Now we see 1, 2, 3, 4, 5, 6, 7 equivalents of CH3COO- and only 1, 2, 3 equivalents of CH3COOH. So now the equilibrium is favoring the acetate, and we have a higher pH. Why? Well, the OH- from the sodium hydroxide removes H plus to form water, and so in order to respond to this perturbation, Le Chatelier's principle says the equilibrium will shift to the right, making more of the acetate ion. We can track this a little more carefully as we add progressively more and more quantities of either the strong acid or the strong base to that initial buffer solution. In all of these cases, we're going to be tracking how the concentration of the A- anion and the HA weak acid are changing, and that allows us to then solve for H+, take the negative log of that, and see the pH change. So here we see some progressive additions of HCl, hydrochloric acid. We start from that equal amount of HA and A-, giving us a pH equal to the pKa of acetic acid of 4.74. But when we add 0.01 moles of HCl, that's going to deplete some of our A- as the reaction shifts towards the HA reactant. So now we have more HA than A-. If we substitute 0.06 molar in to our Ka expression for HA and 0.04 molar in as our A- concentration, then we can solve for H+, take the negative log, and find that the pH is a little bit lower, but not much. We can keep going along these same terms until we have added 0.05 moles of HCl. At this moment, we say that the buffer capacity has been exceeded because those 0.05 moles of HCl have depleted the A- concentration entirely. There's no longer roughly equal concentrations of these two species. We have all HA and no A-. And now the buffer can no longer maintain that same pH that we have before. So buffers are not magic, but they were able to maintain a pretty slow change in pH up until that point. The same goes for adding sodium hydroxide to our initial 1-to-1 buffer solution. In this case, the 0.01 moles of strong base ends up increasing the concentration of A- by that amount, because remember, the addition of base shifts the equilibrium towards the right. It decreases the concentration of HA by 0.01, but these are still the concentrations that we're going to plug into our Ka expression, solve for H+, and we can obtain a pH value by taking the negative log. We see that the pH value increases rather slowly at first until, once again, we have exceeded the buffer capacity by adding so much base that we've completely depleted one of the components of our buffer. If this is starting to sound like a titration experiment, you are on to something. In fact, this is exactly like what happens when you first start to add a strong base to a weak acid solution. There might be a moment along the way where, for example, you have 1, 2, 3, 4, 5, 6, 7 equivalents of acetic acid and 3 equivalents of acetate. This is as though you have added some strong acid to a buffer solution. There's also a point along the way where you have equal amounts of acetic acid and acetate. This is that magic moment halfway to the equivalence point where the two weak acid and conjugate base concentrations are equal to one another. So this is your initial buffer solution before you have perturbed it in any way. And then if you were to keep adding strong base after that point, you will once again end up with the acetate conjugate base in excess, as shown here with the 1, 2, 3, 4, 5, 6, 7 acetate equivalents to every 3 acetic acid equivalents. We can see that there are large quantities of both the weak acid and its conjugate base present, and so the pH changes relatively slowly in this region that we call the buffer region. When you actually add enough base such that the buffer capacity is exceeded, that's when you are able to reach the equivalence point. As I mentioned a few slides back, there might be various motivations to buffer your pH at different values. And so if you would like to create a solution with a relatively consistent pH of 10.6, then you are looking for a weak acid that has a pKa of 10.6. In this case, that would be methyl ammonium. And so you can look up the pKa values for a variety of weak acids out there on the internet, and that allows you to tune your buffer to whatever pH you are aiming for. It turns out that buffer solutions are very important to living organisms because they do not tolerate quick changes in pH very well. In particular, the hydrogen phosphate and carbonic acid buffer solutions are very relevant in our own bodies for maintaining the pH of our blood. It turns out that the normal pH of arterial blood in humans lies somewhere between 7.35 and 7.45, and we would really like to keep it that way. And so in order to keep arterial blood at this pH, we have a buffer solution made up of carbonic acid, H2CO3, and its conjugate base, bicarbonate, HCO3-. As you notice here on the slide, carbonic acid is also in equilibrium with carbon dioxide, which can dissolve in the water that makes up our blood to create carbonic acid. This can be kind of a problem because reactions in cells that transfer energy to our cells, such as cellular respiration, produce CO2 as one of their products. And so this CO2 increase on the reactants side here will dissolve in the blood to make carbonic acid. That increases the concentration of this middle species, which by Le Chatelier's principle is also going to shift this other equilibrium towards the H+ cation. Now, our arteries don't like that. They don't want an excess of H+ turning our blood into acid. This is especially a problem when you are exercising and your cells are demanding a lot of energy from you and you are producing a whole lot of CO2. This runs the risk of turning your blood acidic by producing an excess of H+. However, you do not need to worry about stopping exercising in order to preserve the pH of your blood because your body already has your back. When you exercise, you breathe really hard because your body is trying to exhale all of that extra CO2. When you breathe out CO2, you are removing it from the system of your body. Removing this reactant shifts this first equilibrium towards the reactants, which decreases the amount of H2CO3, which then shifts this second equilibrium towards the reactants as well. And so yes, your body is producing excess CO2, but by exhaling that CO2, the carbonic acid bicarbonate buffer solution can still keep your blood pH under control. Another really useful application of buffer solutions that is not quite as life and death is the use of buffer solutions as acid-base indicators. Acid-base indicators are substances whose weak acid form has a different color than their conjugate base form. So all of the substances shown here have the colors shown when they are in their weak acid HA form, and then they have a different color when they are present as their conjugate base. The pH at which the transition between these two colors occurs depends on the particular substance. So for example, methyl red changes from looking red to looking yellow at around a pH of 5.1. Now, when you go from having an excess of HA to an excess of A-, there is of course a point right during that transition when HA's concentration is equal to the A- concentration, which is that buffer solution that we have been talking about all lecture long. So when those concentrations are equal, that's when you have your color change. That's also the moment where the concentration of H+ is equal to Ka. If you take the negative log of both sides, this is the moment where pH is equal to pKa. So if you are looking to indicate a certain pH to yourself, well, look for a colorful acid-base conjugate pair with a pKa of that same value. Here is a summary in just a slightly different form for many of the substances from the previous slide, showing you the color of their weak acid form, the color of their conjugate base form, and then as you can see, the pH at which this color change happens is indeed the exact same as the pKa of that weak acid. And then there's a little range over here over which you can start to observe the color change. It's not instantaneous, kind of starts to happen a little bit lower and continues over into a higher pH as well. But this can be really useful if you don't know the pH of your solution. You can just add a little bit of, say, methyl orange to your unknown solution. If you were to see it turn yellow, then you would know you have an excess of the A- form of methyl orange, which indicates to you that this solution has a pH above 3.5. You could then take another sample of that solution and add a little bit of phenolphthalein to it. If it remains colorless, that tells you that there's an excess of HA, because HA of phenolphthalein is colorless, which tells you that the pH of your unknown solution is below 9.4. So with these two indicator tests, you know that your pH is somewhere between 3.5 and 9.4. So maybe you take a third sample of the solution, add a little bit of bromothymol blue, and you see it turn green. Green is, of course, what you get when you mix blue and yellow together. So you are at some pH where approximately equal amounts of HA and A- are present. Well, that occurs right around 7.1, which is indeed between 3.5 and 9.4. So just to hammer this point home one more time, the truly important quantity here is in determining the color of your indicator solution is the pKa of this indicator. Because if you have a pH equal to that pKa, you will have equimolar concentrations of HA and A-. You're going to see whatever color is made when those two colors of HA and A- mix together. If instead you observe the color of the HA species, that tells you that the concentration of HA is higher than the concentration of A-. That must mean that the reactants in this equilibrium are favored, which indicates to you that maybe there was an excess of H+ that pushed this equilibrium towards the reactants by Le Chatelier's principle. That excess of H+ indicates an acidic solution with a nice low pH. The opposite situation is when you look at the indicator and you see the color that the A- species has. That indicates that the concentration of A- is greater than the concentration of HA. That indicates that this equilibrium is lying towards the products, which maybe suggests that some excess hydroxide reacted away the H+ to shift the equilibrium towards the products. That excess of hydroxide makes for a basic solution with a nice high pH. A very common application of a pH indicator is to track a titration, because of course what you are looking for when you are titrating acid with some known quantity of base is you are waiting to see when the equivalence point occurs. And unless you have this graph in front of you, it might not be abundantly obvious. When it comes to titrating a strong acid with a strong base, you know that the pH of the solution at the equivalence point is equal to 7, and so it may be prudent to use bromothymol blue as your indicator because its color change occurs at a pH of approximately 7. However, when you are titrating a weak acid with a strong base, as we discussed during our review at the beginning of this lecture, the equivalence point for this type of titration actually is at a pH slightly greater than 7, in which case maybe phenolphthalein would be a good indicator to use because its color change occurs at a pH a little bit higher of 9.4. Notice that this is distinct from the weak acid conjugate base equimolar moment that happens for the acid you are trying to analyze, right? So that equal concentration of, in this case, acetic acid and acetate occurs not at the equivalence point, but at the halfway to the equivalence point. And so these are all the same concept as one another, but importantly, they occur at different points because the Ka values for acetic acid, bromthymol blue, and phenolphthalein in this example are all different for one another. And so even though these are all conceptually the same idea, you do want to make sure that you are selecting your indicator for the equivalence point, not for the halfway to the equivalence point. You may be inferring from the previous slide that there is actually an alternate way to create a buffer solution, where you start from just a solution of the weak acid. So here is a solution of acetic acid. Looks like I forgot my little subscript 3's down here, but you can imagine that this is the concentration of acetic acid greater than the concentration of acetate initially. Now, if I want to make a buffer solution of acetic acid and its conjugate base acetate, I could just add acetate, or I could get to my halfway equivalence point by adding a half equivalent of NaOH, in this case 0.05 moles. At that point, the hydroxide will react with half of my acetic acid, turning it into acetate, and now I have equal concentrations of acetic acid and acetate, just like I wanted for my buffer solution. So it doesn't matter how you get to this point, whether you do it by adding these two species from the get-go or by adding one and then reacting it to produce the other. As long as you end up with equimolar concentrations of a weak acid and its conjugate base, you have prepared a buffer solution. So now you know two ways to go about doing so. At this point, let's go ahead and test your understanding of the buffer solution concepts as applied to acid-base indicators. The indicator for this question is bromthymol blue, which we saw on previous slides looks yellow in its weak acid form and blue in its conjugate base form, with a Ka of 7.9 times 10 to the negative 8. I want you to go ahead and pause the video and take a moment to consider what color you expect to see if bromthymol blue is added to a solution with pH of 13. Welcome back. I hope you have made your selection. Remember that it is the pKa value for an acid-base indicator that dictates where the color change happens. So we have the Ka value. We can take the negative log of this value to get the pKa, which for bromthymol blue is approximately equal to 7. So when the pH of our unknown solution is 7, we will have equal amounts of yellow and blue, and the solution will look green. However, we do not have a pH of 7, we have a pH of 13. This high pH indicates an excess of hydroxide ions. Those will react with the H+ ions that are produced by the acid dissociation equilibrium, shifting that equilibrium even further towards the products by Le Chatelier's principle. So that high pH indicates that mostly A- is around, so the solution will look blue. Another real-world example of colorful acid-base indicators is the anthocyanin compound that is found in many berries and flowers. Anthocyanin looks red in its weak acid form, Ha, and looks blue in its conjugate base form, A-. And so depending on the pH of the soil in which these berries and flowers are grown, you can end up with either a majority of HA molecules around, making the berry or flower look red, or a majority of A- molecules around, making the berry or flower look blue, or if you're at some intermediate pH, you might have a mixture of blue and red, making the berry or flower look purple. This is an experiment you can actually do yourself if you happen to have any of these berries on hand. You can place them in vinegar, which is acetic acid. That will lower the pH, and you should see what color they appear after that. Or you can place them in a little bit of glass cleaner That's ammonia, which is a weak base. And you can see what their color appearance is after you raise the pH in that manner. So the key idea for lecture number 30 is that buffer solutions are made by mixing equal large amounts of a weak acid and its conjugate base. These are useful because they're able to resist large shifts in pH. Specifically, if you want to select a buffer in order to maintain a certain pH, you should choose the buffer whose pKa is equal to the pH that you want to maintain. This is very important for maintaining the pH inside living organisms, but also for maintaining the pH for other important things like food and drinking water. So thinking about the environmental contaminants in drinking water is going to be the subject of our next two lectures.